Introduction. Extraction consists of the isolation and purification of DNA molecules and is based on the physical-chemical characteristic of the molecule. But what is DNA? DNA is a molecule contains the genetic instruction and uses development of a functioning of a living organisms. DNA is made of the strand nucleotides to joined together to form a double helix. Nucleotides consist of sugar, a phosphate group, and a nitrogen base, adenine, guanine, thymine, and cytosine. A phosphate group, negative charge are polar with a gives DNA, and a negative charge makes a highly polar characteristic that is explored for extraction. The strawberry, like the rest of the living beings, also has DNA. And with the following experiment, we will extract in a see in with our eyes.
Material for strawberry DNA extraction. This detergent, 5 grams of the table salt, 5 grams of the meat tenderizer, substitute natural pineapple juice or excretion fresh papaya seeds, 96 alcohol, 100 grams of the strawberries, glass jar, test tube, agitator, mortar, substitute for a blender, and strainer.
Group 3 experiment. Strawberry DNA extraction. Continues. So we're going to get DNA from strawberry with our materials to use and the strawberry without the top because it's difficult to break the leaves. The first thing we will do is macerate the strawberry. For this, we put the strawberry in a bag in order to break the cell walls. A paste as small as possible should we obtain with the help of Wonder Mullet. The paste is placed in a container with the help of a spoon. Continues. So we're going to get DNA from strawberry with our materials to use and the strawberry without the top because it's difficult to break the leaves. The first thing we will do is macerate the strawberry. For this, we put the strawberry in a bag in order to break the cell walls. A paste as small as possible should we obtain with the help of Wonder Mullet. The paste is placed in a container with the help of a.
Hi, I am Montserrat Molina. In the second step, we need to strain the strawberry, leaving only the liquid on the beaker. Then, we need to add detergent. The detergent helps to break down the phospholipid bilayer of plasma membranes and nuclear phospholipid bilayer of plasma membranes and nuclear envelope, which will cause the long DNA-associated proteins to broken down into smaller chains and separated by the detergent. Then, we need to shake vigorously. This procedure ensures that the detergent acts on the entire contents of the extract and not only on the parts in contact with the beaker. Finally, we need to add salt and meat tenderizer. The salt helps to separate the DNA from the stones and is what helps the DNA to precipitate later.
As the sixth step, we measure 20 milliliters of ethyl alcohol that we will add later in test tube that contains the previously prepared mixture. Subsequently, we proceed to pass the mixture that we have to a test tube to be able to better observe the detachment of DNA from mixture. Finally, the alcohol is added to the test tube and observed. Alcohol is used to precipitate DNA because as is soluble in water. When it is in alcohol, it unwinds and precipitates at the interface between alcohol and water.
I'm Caitlin Marroquin. As a result of this experiment, we observe a liquid supernatant that is turbulent. This contains the DNA of the strawberry.
What happened? First of all, it is important to know that strawberries have a lot of DNA, and this is because they are octoploid. During the procedure, there are four important steps. The first step is to freeze the strawberry because when they freeze, small ice crystals form inside each strawberry. These crystals break the cells, thus allowing everything contained in the strawberry to come out. The second important step is to add the meat tenderizer because it contains proteases, that is a protein specialized in the degradation of other proteins, which prevent DNA from being degraded.
The third important step is the when adding the salt. When salt is added to the sample, the positively charged sodium ions they are attracted to the negative charge of the DNA. Neutralizing the charge of the DNA allows DNA molecules to stick together instead of repelling each other. As a result, the repulsion between the DNA strands will decrease. And finally, the fourth step, I think the most important is the addition of the alcohol because this allows us to visualize the DNA because adding alcohol precipitation the DNA given it is insoluble in high concentration of salt and alcohol, and this is how the precipitated DNA for fine wine turn visible straight and the separation limits of the alcohol phase while the rest of the substance remain dissolved.
Conclusions. Observation of DNA in strawberry without optical instruments has been shown to be possible using common materials. This same technique could be used in other tissues, like animals or plants, and demonstrate if it is observable, which is the subject of the project. Strawberry isolated DNA can be easily collected as it is an alcohol-insoluble molecule.
An advantage of DNA was produced because ripe strawberries were used and they produce enzymes, pectinase and cellulase, which are chemicals that aid in the breakdown of cell walls, in addition to the property of creating nine copies of different DNA chains that is then are octaploid.